I welcome everyone to TFT 412 Milk and Dairy Technology class. I am Dr. Olufunke Ezekiel, and I'm your main course facilitator for TFT 412 class, Milk and Dairy Technology. Introduction. Once again, I want to welcome you to this course, Milk and Dairy Technology class, and it gives me great pleasure to welcome you as eager students who are ready to learn the basic principles of those delicious milk products you come across almost on a daily basis. The knowledge and skills you will gain in this course will afford you the opportunity to learn how milk is produced, as well as the manufacture of various milk products, such as cheese, yogurt, butter, ice cream, and so on and so forth. I know you can give me a lot of more examples, even more than I could imagine in my own little brain. In this course, you will also learn the process variables and various unit operations that are involved in milk processing, right from reception to final products. This course will prepare you to work either as a production or quality control personnel in milk processing industry. You will be exposed to both virtual and practical hands-on experience as it relates to milk processing and packaging as well. As we move on in this course, you'll be distributed into 4 to 5 groups every 2 weeks for effective practical laboratory activities throughout the duration of the course. Milk and Dairy Technology course teaches the fundamental of milk, milk properties, milk composition, milk handling from reception to final products. It also teaches milk transportation, milk storage, milk quality parameters, and production of several products from milk. In this course, the various principles and fundamental mechanisms of manufacturing these products will be described, as well as the associated unit operations and processing units. We'll talk about sanitation, as well as management of the generated waste. In this university, it is expected that at 400 level, you would have acquired some knowledge of food science techniques, such as food processing skills, while in 300 level, and therefore should be able to operate some food processing units and equipment effectively. So this course involves both the practical session and the theory. The objectives of this course is to enhance your capacity in food processing and preservation, especially as it relates to milk, with specific emphasis on milk and milk products. I hope you are ready to learn and put to practice what you will be taught, because the key to milk processing and production of milk products will be placed into your hands, and you are expected to put in your best as the time to do your work is placed at your disposal. So thank you for listening to that introduction. So briefly, we'll move to the course outline. What are the things that you are expected to learn? Number one, we'll talk about milk overview, which we will deal with in the first module and session 1. We'll also deal with fluid whole milk, have the otherwise referred to as pasteurized homogenized milk. There is also evaporated milk. We also have sweetened condensed milk. We have dry milk powder in module 5. Module 6, we'll talk about ice cream, how it is produced. We have lollies, ice lollies. Those ones they are used to, especially when you were young. They put it inside a stick. I hope you can remember. Then we have butter. Butter, yeah, some people used to mix up butter with margarine. Butter is from milk, while margarine is from fat. I hope that is noted. We also talk about fermented milk products, with specific emphasis on the very popular ones. Talk about yogurt, and also we'll talk about cheese. The one that is popular in Nigeria is the soft cheese, usually referred to as wara. So then, in continuation of course outline, we will deal with dairy waste management and processing, how we can add value. There are some ways that we can actually add value to. For instance, whey. We can add value to whey, make value-added products from it, recover some of the proteins which may be added into other food products. Then we'll also talk about dairy plant sanitation. How do we keep the dairy plants neat? How do we keep it away from bacterial contamination so that we don't get our products contaminated? So today we'll be talking about module 1, and session 1 will deal about milk overview. And this is the outline for session 1. We have introduction. We'll talk about milk and its components. We'll also talk about milk chemistry. We'll talk about milk microbiology. What do we have up there? Anybody? Anybody? Can unmute to just contribute. There's a
milk in a cup.
Milk in a cup. Thank you. Forming a splash. Thank you very much. What exactly is milk? Yeah. Milk is the lactate secretion from the mammary gland of mammals. Milk is the lactate secretion from mammary glands of mammals. But when you look at milk, it's in various settings because of the various variability of milk. We have different composition depending on which animal it's coming from. And also because of the importance of milk in the milk industry, there are legal definitions available also for milk in order to set a legal standard. So in some other clime, milk is defined as the lactate secretion practically free of cholesterol obtained from one or more healthy mammals. When we talk of the animals, it has to be mammals. Animals that can give birth to young and also give suck. Usually cow. In our setting, we have it usually cow, and it must contain nothing less than certain percentage of milk, milk solid. At times we have 1.25% of milk solid, as well as 1.25% of milk fat. Now, these are examples of sources of milk. We have the first picture talks about cow. We also have goats. You can get milk from goats. We can also get from mare. Mare is the female horse. Then we can also get from lamb. Lamb is a variation of a variation of camel. And of course, we can also get from humans as well. Let me pick this and I want to adjust this. Yeah. So we can also get from humans. In some areas in the world, they're already collecting human milk, even for storage, especially for mothers that may not be able to breastfeed for one reason or the other. But we are not yet there now in Nigeria. But I know in developed countries, they now have storage where a cold storage where they keep human milk. So essentially, milk is the first food of the young, and it is a complete meal. When I say complete meal, it contains all the nutrients that is associated with food, and it has a high biological value, of 0.75 to 0.8, and that makes it a very good protein source. And also because it's a complete meal, it is also highly susceptible to microbial contamination. So we have to note it that we cannot just leave milk immediately a calf has been born, and you are able to wait for some days, able to milk the mother. We cannot leave milk on the ground. It must be processed immediately or kept cold in a cold storage. There are several ways in which we can describe milk. One, milk can be described as an oil in water emulsion. When we talk about oil in water emulsion, it means that droplets of fat are dissolved. They are in the liquid, in the water, to form emulsion. So water will be the dispersing phase, and oil will be the dispersed phase. Also, milk can be described as a colloidal suspension. Of course, it is obvious milk is not a clear liquid. There are some things, some materials, some nutrients that are suspended in it, which are not they are not soluble. So it makes it it forms a colloid. So that's why we have milk that is not clear in appearance. And then lastly, milk can also be described as a solution. Why solution? There are also some materials. A very good example is lactose that is dissolved, is the milk sugar. It is actually soluble in milk. So those are several ways in which we can describe milk. What are the principal components of milk? We have water, solid, not fat, fats in dry matter, carbohydrates in form of lactose. Of course, we also have oligosaccharides, but it's not in significant quantity. We have protein, ash, or mineral substances. We also have organic acid, vitamins, and then we have some we can refer to as miscellaneous. Examples are we have immunoglobulins, we have antibodies. These are very important also. But of course, during processing, we may lose some of them. Now, we cannot have we can only have ranges for this composition because the principal components of milk, they vary. They vary from one animal to the other, even though they may even be the same species. And that's why during processing, we have an issue of blending so that we can reach particular maybe fat content, particular water content. So all those ranges are as highlighted above. Definition of terms. We cannot run away from describing milk fractions. Milk as it has several fractions. So from time to time, we come across some of these terms. So it is better to define them right from the onset. We have plasma. There's plasma. Plasma is milk minus fat, and it is usually called the skim milk. Of course, not the all content milk, fat content may not be removed, but a larger percentage would have been removed. We also have serum. Serum is plasma minus casein micelle. Casein as a type of milk protein, but that is not the only protein that is in milk. We also have whey protein. So when you have plasma, it's actually the whey protein, the liquid whey that is called serum. Plasma minus casein micelle will give us serum, usually referred to as whey, liquid whey. We also have solids, not fat. In other words, we have some solids that are referred to as fat. But when we want to break it down into fractions, we consider fat separately. We also, that is milk fat, we also consider solid, not fat, which is made up of proteins, lactose, minerals, acids, enzymes, and vitamins. Then finally, when you talk about total milk solids, we are actually referring to fat plus the solid, not fat. Colostrum, yeah, it's we'll come across colostrum as well as one of the terms. This is the milk produced in the first 2 to 10 days postpartum. And it is very pertinent to talk about colostrum because it contains antibodies, which is usually utilized by the young cow. So for commercial purposes, it is usually advised that the mother cow or the animal that is we are obtaining the milk from, we do not milk it in the first 2 to 10 days. In fact, some people extend it to 15 days. Why? So that the antibodies will be available to the young cow, the young animal, so that it can be able to resist diseases and or what have you that may want to challenge the growth of the young animal, the young mammal. And it is usually different from milk. It has strong odor. The taste is usually bitter as well. And not only that, the color is usually reddish to yellow color. It means it contains a lot of vitamin A. You know, remember that's part of the constituents of milk. Then it has large percent of immunoglobulin, which is very important as an antibody and also to fight against diseases and reactions. However, it is richer in other milk constituents, except lactose, potassium, pantothenic acid, vitamins, and water. It is usually more viscous than the milk that is usually used for commercial purposes. So let's note it. Colostrum is obtained in the first 2 to 10 days, and in some other clients, they are advised to extend it to 15 days before you begin to collect the milk from the mother mammal for commercial purposes. Milk structure. You have milk structure being, let me say this, it shows. Okay. The milk structure, the structure of milk. As we see milk, it contains several components. And when you look at the structure under a microscope, you can see several other things that are not even visible to the eyes. Milk, you can see at the right side of the screen, we have about figure 1. On figure 1, we have about 3 separate magnification of microscope. The first one is 5 times 5. Then the second one, B, the region of B is times 500, and then the region of C is times 50,000 in terms of magnification. So we have there is the milk plasma, there's casein micelle, we also have milk serum. We have milk serum and lipoprotein particle cells. You can see that the lipoprotein particle cells are not, the fat globules are not very visible at times 5 and times 500, while at times 50,000, you can see casein micelle, serum, membrane, and fat globules. We also have milk chemistry. Milk lipids contains mainly triglycerides. We also have glycerol, which is the backbone binding up to 3 different acids. There's fatty acids made up of hydrocarbon chains. We also have other milk lipids, which include phospholipids, cholesterol, free fatty acids, and glycerols. Under milk chemistry, we have milk protein. About 95% of the nitrogen in milk is associated with protein. And these are broken down to 75% casein, which is equivalent to 80% of milk protein. And we have serum or whey proteins, as earlier mentioned, 18%. Enzymes, mainly hydrolysis, and miscellaneous proteins, antibodies, 2%. Hydrolysis in milk include lipoprotein, plasmin, alkaline, and phosphatase. These are important, especially alkaline phosphatase. These are important enzymes, which indicate quality of milk. We also have milk carbohydrates, lactose, which is made up of glucose and galactose. There is also glucose on its own, galactose on its own, and then we have some oligosaccharides making up the milk carbohydrates. Milk microbiology. Milk on its own is sterile. It is sterile, and at secretion, as it is coming from the udder, it is sterile. But it is highly susceptible to microbial contamination through handling the hairs on the animal. Then as it's coming even through the udder, it can be contaminated, the materials used, to collect it. So it can be contaminated by bacterial coliforms and mold. In summary, milk is a complete food, and the major composition of milk is water. That's about 80 to 85% of water. And you can also see that milk is an emulsion. It's fat in water emulsion. And unless contaminated, it is sterile. Raw milk, unless rapidly processed, spoils quickly. However, milk processing may lead to some losses too, from your previous knowledge in year 3. You can see that milk, when processed, will lose some of its nutrients. For further reading, we have Dairy Technology, Principles of Milk Properties and Processes by Peter Westra and et al. We have Processing Technologies for Milk and Milk Products. You're expected to consult some of these texts. And also there's a link to internet-based reading. You can see it also on the University LMS as you connect. We have a small class activity. There'll be distribution of the class into 2 groups for a debate. Group A will defend consumption of raw milk, and Group B will defend consumption of processed milk. We also have discussion forum, which will be completed on the university's learning management system. Some of you need to still sign up on this. I think I've sent out the notice. So the question is, if given the opportunity to choose between raw milk and processed milk, which one would you prefer to consume? And kindly give reasons for it. Thank you. That's the end of my lecture for today. In the subsequent week, we will be talking about milk testing, milk grading, and some of the materials that we use for testing the microbial quality of milk. Thank you very much. It's been a pleasure having you around. I don't know whether there are questions or observations from my students. Any observation?
Thank you very much, Ma. I have a question.
Okay, who is speaking?
Yeah, this is Oladayo speaking.
Is that Mr. Timileyin Oladayo? All right.
Yes, Ma. So my question is, you talked about colostrum being milk produced in the first 2 to 10 days. So my question is, does this happen in humans too?
Oh yes, it happens in humans. It happens to all mammals, and that is why immediately a baby is born, you're advised to put the baby to suck, so that the baby can get enough immunity. You know, this is a new environment for the baby, which is no more sterile, like the condition the young mammal is coming from. So it's also it's applicable to human as well.
Thank you.
Okay, you're welcome. So we'll hope to meet again on the platform. As some of you that can log in, the forum discussion will be open for your contribution. Please feel free to express your mind, but of course, especially with reasons. You might have gone through some of the text. So see you again next week when we'll be having another class, same time, online. So at this point, I want to say a very big thank you to every one of you. Okay, Mr. Ezekiel is raising up his hand. Okay, can you please unmute? Thank you. Yes, can we hear you, sir? Mr. Ezekiel, kindly mute your microphone. Thank you very much. It seems your area is a bit noisy. So thank you. You can,
hello.
Yeah. Hello, sir. Yeah.
Can you describe the best way to handle milk to prevent contamination?
Well, immediately after milking a cow, the best thing is to take it straight for cold storage. And not only that, the hoodah of the mammal must be sanitized. In some instances, you can shave a little aspect of the hair away, because if the hair also enters, it's a form of contamination. You can also be careful the kind of treatment you give the animal, because somatic cells can come in from infected animals. So keep the animal healthy, keep the milk collecting equipment, keep them neat, and make sure the environment is clean. Then the handler too, should be neat and well sanitized, equipment well kept. Okay, in the absence of any other question, thank you, sir. I want to say a very good afternoon. Meet you again next class. Thank you, and God bless.